CD38 is a surface marker that's widely expressed on hematopoietic cell lines, specifically with the B cell lineage, but it's also expressed on non-hematopoietic cell lineages as well. It is commonly expressed on plasma cells, and the scientific rationale is there to utilize this as a potential target for therapies, and several anti-CD38 monoclonal antibodies have been examined in early phase clinical trials. The two that are the farthest ahead in terms of clinical development have been the antibody Daratumumab, which is a humanized IgG kappa monoclonal antibody, and Isatuximab, which is a chimeric IgG kappa antibody. And I detailed the monotherapy data with Daratumumab, as well as the data in the relapsed refractory setting. I did the same for Isatuximab. We do have data with the pomalidomide combination as well as carfilzomib combination. So I walked through the phase 1 data, the phase 2-3 data with both of these monoclonal antibodies, highlighting which clinical populations those data will be most relevant for. With the Daratumumab-based combinations, I discussed the data with lenalidomide as well as bortezomib combinations, so namely the POLLUX and the CASTOR trial, highlighting the fact that the three-drug combination with this anti-CD38 in both those trials came up with positive results compared to the two-drug combinations in terms of depth of response, overall response, MRD negativity rates, as well as progression-free survival. And then I also discussed the data that exists with the combination of pomalidomide, dexamethasone, and the most recently presented the CANDOR clinical trial, which looked at daratumumab, carfilzomib dex versus carfilzomib dexamethasone. The populations are obviously different with each of these trials, and so it would be important to bear those in mind when picking which option may be the best for your patient. For example, with patients who would get dara with Lendex, that clinical trial did not have patients who had prior Len refractoriness, but patients with bortezomib or exposure or refractoriness were included. Similarly, with the dara bortezomib dex combination, patients who had prior Len exposure or Len refractoriness were included, but bortezomib exposure or rather bortezomib refractoriness were excluded. So that would be the if you're looking for a non-Len combination, that would be something to consider. More impressive data, however, are with the daratumumab combination with carfilzomib dexamethasone, where the median PFS has not been reached after a 16-month follow-up, whereas that for KD was about 16 months. And about a third of the patients had Len-exposed and refractory disease. So I think that would be clinically meaningful. In the same way, the Isatuximab Omdex combination appears to be very effective. It read out positively for patients with relapsed refractory myeloma, and Len-exposed or refractory patients were enrolled on that study. And the IKEMA data, which was the combination of Isatuximab with carfilzomib dexamethasone compared to carfilzomib dexamethasone, also read out positively. It would be interesting to see what the median PFS is for the three-drug combinations with both these monoclonal antibodies as a potential distinction. The depth of response appears to be similar. The safety profile appears to be similar. So it will be really the clinical benefit that patients get in terms of disease control, which will probably be distinct. And then the route of administration and this schedule of administration. I did not talk about the cost of drug, but if you look at the global care of myeloma patients, I think that probably will also come into play.